Hi, I'm Anna. I'm a first year PhD student in the Henstridge Lab at the University of Dundee, and I'll be giving a bit of an overview about some of the work that I've been doing over the last year. So I thought I would start with a little bit of a background about how I got to the University of Dundee and how I got into motor neuron disease research. I did my undergraduate degree at the University of Glasgow, where I studied neuroscience, as I've always been interested in the brain and how the brain works. I've always had an interest in research and was fortunate enough to be able to do a year of research as part of my undergraduate degree, and I did this research in the Bramham Lab at the University of Bergen. The Bramham Lab studies tiny connections between nerve cells, known as synapses, and specifically they were studying how these synapses contribute to memory formation in healthy contexts. I loved this year. It really confirmed to me that I want to continue research, specifically studying these tiny connections in the brain. When I came back to Glasgow, I finished my undergraduate degree and continued working at the University of Glasgow as a research assistant, this time in the spinal cord group, where I was involved in studying chronic pain and trying to understand how the spinal cord contributes to our ability to detect sensations on the skin, like temperature. Whilst I really loved this research, I knew I wanted to get back to studying synapses, and I wanted to take my experience from the last few years and apply it to a disease context. As while I was interested in how the brain works in healthy conditions, I was also interested in how the brain changes in disease and how we can target these changes to treat patients more effectively. Through this interest, I found Chris Henstridge and his lab at the University of Dundee, and I've been fortunate enough that they've taken me on as a PhD student, and I've been here since January. So my research looks at synapses, specifically in the context of motor neuron disease. And synapses are the site of communication between two nerve cells. This can be two nerve cells in the brain, or as shown here, a nerve cell forming a connection with a nerve cell in the spinal cord, and they form a connection that looks like this. There's a huge number of synapses in the brain. If we could count 1,000 synapses per second, it would take nearly 32,000 years to count them all. So there's a massive number of synapses in the brain. And synapses are also really vital for our day-to-day functioning, as they are important and they're made when we form new memories, and they're involved in thinking, learning, and movement. So really, really important. In a healthy brain that weighs around 1.5 kilograms, there's around 1 billion neurons or nerve cells, and these nerve cells can form connections with many, many other cells, resulting in around 1 trillion synapses in a healthy brain. What research has found is that in the early stages of motor neuron disease, there's a loss of these synapses, and this is found to happen before symptom onset or loss of nerve cells, suggesting that it might be an early event in motor neuron disease that we could target to try and delay or prevent progression of the disease. Loss of synapses is also a progressive event, as we see a more severe loss later in the disease, again suggesting that if we can target it earlier in the disease, we might be able to delay this loss of synapses, which might help to improve patient welfare. However, the mechanisms and how this synapse loss occurs isn't completely understood, and that's where my research comes in. I'm investigating the when and why of synapse loss in motor neuron disease to try and find new methods to treat the disease and improve patient welfare. So in the lab, we use several different model systems to investigate this research question, and the first of which we use is Drosophila melanogaster, more commonly referred to as the fruit fly. And the first question I always get when I tell people I use fruit flies in my research is why? Why would you use flies to study a human disease? But there's actually a huge number of benefits to using fruit flies in neuroscience research. First, flies have a short lifespan of just under 2 months, and this is important, particularly when we're studying progressive diseases like dementia or motor neuron disease, because we can track progression of the disease in a relatively short time frame and get an understanding of whether our interventions might help to delay or prevent onset of certain symptoms or changes in the brain. Second, flies have a short developmental time period of just under 2 weeks, and female flies can lay up to 100 eggs a day. And this means that we can study a huge number of flies in a short time frame to increase the robustness of our results. Finally, around 60% of proteins that have been found in humans are also found in the fly, and around 75% of proteins that have been implicated in human disease are also found in the fruit fly. And this is important because it means we can study these proteins in more detail to try to get a better understanding of how they contribute to disease and how we can target them. So now we know why we would use fruit flies in our research. The next question is, how do we use them to study motor neuron disease? And in the lab, we have flies that have been scientifically modified to carry proteins important in motor neuron disease. For example, TDP43, a protein that has been implicated in around 97% of cases. These flies will exhibit movement problems and also changes in the brain that might be similar to some of the changes we see in the disease. And we can use these flies to study whether any of our interventions might help to improve the symptoms or changes in the brain. And we can do this using several different methods. First, as motor neuron disease is a movement-related disorder, we can study the movement of the flies by using what's known as a climbing test. We can test whether the ability of the flies to climb up a vial, shown here, is affected and whether any of our interventions might help to improve this. And we also have infrared beam technology that we can use to study their movement on a longer timescale. Secondly, we can study cell death to see when this occurs in the flies and if, again, any of our interventions might help to delay or prevent the cell death. Finally, and what might seem a little bit left field, is we can use the fly eye to study breakdown caused by proteins known to be important in motor neuron disease. And this is because in the fly, we can make sure that these proteins, like TDP43, are only carried in the eye of the fly, and this causes significant breakdown. On the left here, we see a healthy fruit fly eye. The bristles or hairs are very well organised. There's a nice red colour, and the eye overall looks very healthy and organised. If we introduce TDP43 into the eye, we observe significant breakdown of the eye as shown in the middle here. The bristles become very disorganized, we lose some of that red colour, and overall the eye just looks very unhealthy. And what we want to do is investigate if we change different levels of proteins in the synapse or components of the synapse, can we help to restore the eye to look more like the eye on the left? And that's what we do in the lab. For example, on the right here, we've reduced levels of a protein and have found that while the eye doesn't look exactly like the healthy eye on the left, it does look much healthier than the eye in the centre. We have a lot of that red colour back in the eye and it looks a lot healthier. So we can use this to study whether altering different levels of proteins or components of the synapse might help to improve breakdown caused by proteins implicated in motor neuron disease. Another model system that we use in the lab are stem cells to study cell damage in motor neuron disease. And stem cells are a hugely powerful technique in science as they can be prompted to grow into various cell types found within the body. We can take stem cells that have been donated by patients with motor neuron disease and drive them to become nerve cells to investigate changes in these cells compared to people without motor neuron disease and understand if we can target these changes to improve the survival of the cells. So we take cells that have been donated by patients with motor neuron disease and we can encourage them to grow into motor nerve cells, which are the primary affected cells in motor neuron disease. But what we're also doing is encouraging them to grow into sensory nerve cells. And these are cells that help us to detect sensations on the skin, such as touch, temperature, or pain. Interestingly, what research has shown is that these sensory nerve cells appear to be largely protected from the damage observed in motor nerve cells, and therefore we want to compare these cell groups to understand what the protective mechanisms in sensory nerve cells might be, and if we can translate these to our motor nerve cells to increase their survival. Once we have our cells, we can study the cell damage and the differences in the cells through different mechanisms. We can measure cell death again, and here we can investigate whether any of the protective mechanisms observed in the sensory nerve cells might help to increase survival in motor nerve cells. But we can also do what we do in the flies by investigating whether any of our interventions or altering levels of different components of the synapse help to increase survival too. We can also use a microscope to study synapses to investigate when these might be lost in the motor nerve cells and if our interventions might help to increase their number or change the composition of them. This is a very powerful technique as it gives us a human-relevant model of the disease and might identify new methods to increase cell survival. So, overall, I hope that I've highlighted that by using data that has been collected from human patients highlighting this loss of synapses, we can use our model systems to gain a better understanding of the when and why of synapse loss in motor neuron disease, to ultimately understand this phenomena in more detail. Not only do we want to understand these events, we ultimately want to use these model systems as a platform to test new drugs and test their efficacy to get a better understanding of whether they may be effective in patients and might provide a more effective treatment. So, overall, through understanding and targeting the early steps of human disease, we want to understand the where, when, and why of synapse loss and how can we stop it. So with that, I would just like to thank the Henstridge Lab in the University of Dundee for allowing me to complete my PhD here and for their incredible support and advice so far, as well as the McGurk Lab and the Hales Lab for their help and advice with the flies and stem cells respectively. I'd also like to thank our funders and would like to thank you all for listening.